Good afternoon, everyone. Thank you to Tim and the organizers for putting on a great event. It's always fun to come back to the Mesa. I'm Sarah Meeks, and I'm the Senior Vice President of Business Development at MaxCyte, and my team is responsible for putting partnerships in place with therapeutic product developers. I think I'm going to try to do this. Oops, next. So I'm going to tell you a little bit today about partnering to build your next generation of cell therapies at MaxCyte. I want to talk to you about our partnership model, what it entails, what the value we bring, how we're going to work with you and work very closely with you. We have over 600 platforms in place, and our technology unlocks significant potential in advanced therapeutics. We're supporting next-generation cell therapy programs and innovative non-viral engineering approaches. And our innovative and powerful business model is focused on value creation, thus enabling your success. We have 23 partnerships in place to date, including 5 signed this year with Catamaran Bio, Walking Fish, Vittoria, Lyell Immunopharma, and Prime Medicine. Who are we at MaxCyte? The company was formed in 1999. Where our FDA master file was filed in 2002. It's over 20 years old now. We listed on the London Stock Exchange in 2016, introduced our current platform, the Expert platform, in 2019, listed on the NASDAQ in July of 2021, and just last year moved into our new headquarters, had our grand opening in Rockville, and brought in our manufacturing in-house. Critically important to us to support clinical and commercial phase of our partners. We are collaborative, innovative, and experienced. We also understand the industry challenges as presented today, and this is to say that there aren't going to be some in the future that we're all going to have to tackle, but presently there's a lack of industry standards for process design that can cause costly delays and inconsistent results. Our technology allows plug and play with rapid optimization, delivering reproducible outcomes and the ability to seamlessly scale up from pre-IND to clinic and commercialization. The next generation cell therapy programs are often and increasingly quite elegant, complicated, complex, with multiple edits potentially. It's going to move even probably more complicated, and that is probably the promise and the innovation that's going to occur in this field that we all are going to need to keep track with. Our platform facilitates multiplex and sequential engineering approach without payload or capacity limitations that viral vector approaches may have. And then finally, regulatory risk increases with new unknowns, donor cells, next-gen approaches, new indications. And as I said, our master file at the FDA was put in place in 2002, and it can be referenced in regulatory filings to accelerate and de-risk regulatory review. So now a little bit about partnering with MaxCyte. So let's build better cells together. What if you had a really complex pipeline with multiple programs, perhaps multiple indications, multiple targets, and those programs are supported by, let's say, over 20 clinical instruments across multiple countries and multiple CDMOs. And this is only in clinical phase. Imagine then what it would be in commercial phase. Would it be of value if you had seamless optimization from research to phase 1 to BLA filing? What if there were 5 functional teams to support you on call? You had audit-ready quality support. You had partnership reviews, a really strong alliance management with one point of contact to facilitate everything that needs to happen around the globe to bring your clinical programs and across the globe and into commercial phase, potentially across the globe. There was seamless maintenance and upgrade coordination. And you had 24/7 tech support in place and ready for commercial phase, all along with 20 years of regulatory experience. I'm wondering if that would be helpful. At MaxCyte, we think it would, and our joint goals at Partners is alignment on this, on creating value and enabling your speed to market. And we really think about it in terms of these 6 pillars. It's not just the technology or the instrumentation, it's the technical support. It's the regulatory support, it's the quality systems in place, it's our global partnership support, and it's the operational improvements that we've made to ensure that your clinical and commercial needs are met and not backordered and are ready for you. In the next couple of slides, I'm going to dig in a little bit deeper on these. Our FAS and field engineering teams, this team has been designed purposely to have comprehensive expertise to support the expanding array of applications. This graph shows across the bottom a number of different areas of expertise in engineering cells, including immunology and gene editing, iPSC and stem cell expertise, process development, cell-based assays. We have groups here at the top of the bar graphs, our PD team, our R&D team in the middle, and our FAS, or field application team, on the bottom. And importantly, what we've done is, while your company may be located here in Southern California, if there is an FAS that has the experience and expertise that lives in, let's say, London, we're going to get that FAS here to help you and to design and to optimize. And so the team is interchangeable, all coordinated also through our headquarters, which has a large R&D team as well as a in-house process development team. We also, as I mentioned, have inclusive and flexible regulatory support. So we've been supported in over 45 clinical trials to date. We've supported those. We have over 20 years of regulatory experience supporting clinical programs. There's flexible global filing to support of an IND or a CTA, BLA, others. And that's either through master files that are in place or associated technical files. Importantly, if you are going into a country that we have not been into yet, our regulatory team is going to support and provide the guidance or the necessary support to be able to file in that country. We have rapid expansion into the APAC countries with in-country regulatory support in many of those countries now, and have had successful filings in numerous countries, including the US, Canada, Australia, and others listed here. Our FDA master file includes all components intended for use in cGMP-compliant manufacturing, including our instrument, buffer, and processing assemblies or consumables. We also have continuous operational improvement and make strategic investments. And in this case, this is a figure, 45%, and this is these were our R&D investments as percentage of our 2022 revenue that we put back into innovation and our science and our build out of knowledge of the science and the innovation that is occurring. We want to stay absolutely abreast or even slightly ahead of the work that's being done in the field so we can support seamlessly and effectively. We can help you build better autologous therapies. A couple examples here. As we know, cell recovery in autologous cell therapies is critical. And so part of what the value that we offer is the ability to produce consistent and reproducible results from donor to donor. And importantly, this is just an example of this is the manufacturing run for a patient that's in Chicago needs to be consistent with the manufacturing run for a patient in London. And because of the support that we provide, and not only the instrumentation, but the support around it, assuring that the CDMOs are up to speed and are trained and are completely comfortable with manufacturing using MaxCyte, we're able to support this. We can help you build better allogeneic cell therapies as well. And as we know, that important to the allo therapies is the ability to have large number of doses from single manufacturing runs. With the volume that you can electroporate with MaxCyte, you can have an increasing number of doses, maximize those doses, which hopefully will eventually would drive down COGS. So this is of value, and we're working with a number of partners where this is critically important. So better cells in action, just a little bit on what we're doing and where they are right now. So one of the areas that the BD team, working very closely with our sales and FAS team, is to drive innovation and early clinical product development in academic and translational centers. We've partnered with over 90 academic and translational centers worldwide. The graph on the left here shows the growing number in the different countries that we're placing instruments with and developing relationships with. With that comes clinical programs, the support and the alliance management support, just as we would provide that for companies. We also do provide that for academic programs. On the right here are the global translational programs that have cross-referenced our MaxCyte master file or an associated technical file, and as you can see, it's increased over the last 5 years, and it continues to increase. So this is important for us to be able to work with innovators and early and train, help them optimize their programs on MaxCyte very early to make for seamless translation and scale up from early clinical to late clinical and ultimately to commercial stage. We're the leading partner for complex cellular engineering. We're supporting clinical and commercial therapeutic product success. We have 23 partnerships that we call them SPLs, or strategic platform licenses. There are over 125 programs in these involved in these licenses, and 16 active clinical programs as deemed cleared IND or equivalent. This data was as of very late last year, so will be updated at the end of this year. As you can see, there are a number of programs with different cell types, with different therapeutic approaches, different indications, and different targets here. Just a brief commentary, and this was given by the CRISPR CEO Sam Kulkarni at the Bank of America Healthcare Conference earlier this year, and really the point is here is he was talking about the ecosystem in and around cell manufacturing, and said that our take home was that really the innovation that's happening around platform technologies is critically important and has been of quite a benefit to therapeutic product developers to bring new safe, efficacious cell-based medicines to patients. So it's really a testament to the innovation that's happening in the sector and really the synergies of those innovations coming together. We have a number of clinical trials. This is a graph showing the clinical trials that are active right now. These are part of our commercial program, the company-based programs, as well as the academic programs. Some of the programs are named on this. They're in the public domain that they're being manufactured using MaxCyte. You can see Azer-cel here from Precision Bio, Trem-cel from Vor Biopharma, Exa-cel from Vertex, both the sickle cell program, which has got a PDUFA date in December, and the Exa-cel program for beta-thal, which has got a PDUFA date for end of quarter next year. We're terrifically excited about all of these programs, and importantly, as you can see, they're in various clinical development, and again, this doesn't include the sort of preclinical programs. There's a robust preclinical pipeline behind this from our partners, both academic as well as industry. We're also building out better cells to reach more patients, increasing in the number of indications. And again, this is a number of indications grouped here by heme malignancies, genetic diseases, infectious disease, solid tumors, increasingly in rare disease. They're not disclosed here. They're in preclinical work. There are a number of gene editing modalities or engineering tools that are enabled in the clinical trials here shown up in the upper right. And again, the first MaxCyte-enabled therapy is expected to be approved late this year, early next, and that would be from Evaluate Pharma, and that's Exa-cel. then finally, just leaving you with a couple words from a couple of our partners. And as here are two autologous-based approach companies, and two that are approaching their products using an allo approach. There are a variety of cell types, indications they're going after, and targets. So thank you. The slides will be available, as I understand, and taped. And thank you. Let's build better cells together.